Hello, everyone, and welcome to today's webinar. I'm Chang Lim, and I'm excited to introduce you to our enhanced developability assessment platform, DEVELOPICK 3.0, which is designed to improve biologics candidate selection. In this session, we will explore how DEVELOPICK 3.0 enables smarter, faster, and more precise assessment. So here's what we will be covering today. First, I will introduce the scope of developability assessment and why it's critical in biologics development. Then I will explore the key factors that impact molecules' developability, followed by an in-depth look at DEVELOPICK 3.0 and how it supports candidate selection. Next, I will go over the analytical method we used in DEVELOPICK 3.0 to evaluate molecule, including biophysical characterization, solubility testing, and sequence-based assessment. And then I will wrap up the key takeaways and Q&A session. As you all know, one of the biggest challenges in biologic development is protein degradation. Various factors such as unfolding, aggregation, and fragmentation can impact the molecular efficacy, stability, and manufacturability. This issue arises due to a molecular inherent properties as well as external stress factors like temperature, pH, and mechanical agitation during processing and storage. Without early assessment, this risk may lead to failure in later stage. Developing enables early detection of these potential issues, allowing developers to take proactive steps to mitigate them. Considering whole drug development process from discovery to commercialization, process development and clinical trials needs to have sizable budgets to proceed. Therefore, it is very important to predict the developability before real process development stage starts to save the money for development. That's why we launched this service for our client. DEVELOPICK helps de-risk biopharmaceutical development by assessing molecular characteristics early and providing risk mitigation strategies in collaboration with our USP, DSP process development teams. By integrating these insights, clients can streamline their development process and reduce the likelihood of late-stage failures. At first, I would like to brief you on our history in making DEVELOPICK. From 2020, DEVELOPICK has been updated from 1.0 to 2.0 and then to 3.0 in 2024. At first, we had thermal stability predictions using TSF DRS system and thermal stability low pH stress testing. Now we have those items and additional solubility, high pH stress, and hydrophobicity testing with good in silico molecular assessment tool. We also have a real solubility testing tool for confirming the feasibility of high concentration for the target molecule. This advancement allows for a more comprehensive assessment of molecular developability, helping developers make more informed decisions. Over the years, we've observed key trends in what our clients are looking for. Over 60% of clients wish to get information about complex proteins like FG fusion protein, bispecific or multi-specific antibody, etc. And 35% of molecules were monoclonal antibody, including IgG1, 2, and 4. In case of concentration expectation, 20% of clients wish to confirm the feasibility of high concentration that is over 100 mg per ml concentration of DSNDP using real solubility testing. This insight reflects the growing demand for the robust assessment tools that can handle increasingly complex biologics. So, how does DEVELOPICK work? At first, we use our own program to evaluate the molecular characteristics based upon the primary sequence information on the instance open website program, which one all know. And we would perform the relative solubility study using PEG assay. Analysis through DSF-DLS system for biophysical characterization is also done to gain insight from the thermal stress perspective like T onset, TM, and T aggregation. After that, we perform short-term stability study to assess real thermal stability in harsh conditions, and confirm low pH and high pH stress study to predict the potential risk for virus inactivation step and pH neutralization step during DSP process. And we also analyze hydrophobicity using HIC method to assess the potential risk during process development due to hydrophobicity. All of these can be done with just 80 mg of protein in under 1 month, providing fast and reliable insight. Let's look at these one by one from biophysical characterization step. At first, we use DSF, DLS, and SLS system to predict the thermal stability and molecular size distribution using the PDI value. On the right side of the slide, you can see the TM and T aggregation graph. And below those graphs, you can also see the DLS data to calculate the PDI value to find out the polydispersity of the molecule. Using T onset TM T aggregation, we assess the molecular thermal stability characteristics, and using DLS, we can confirm the possibility of self-association or aggregation propensity. If our client wishes to confirm the colloidal stability on specific buffer type and pH condition, then we can also perform the study during the developing period. And this is optional service for DEVELOPICK with also delta G measurement to assess the chemical stability using the Hunke system. If our client use this molecule, then they can select the preliminary pH buffer conditions for their molecule and get more information about the stability. Here is one example of the optional biophysical characterization. On the left side, you can see various color on the graph for diffusion coefficient. If the molecule shows a positive KD, which corresponds to the sky blue line, the molecule repels each other. If the molecule shows a negative KD, which corresponds to the light green, the molecules are attracted to each other. So that means the molecules are more likely to aggregate during PD or stability study period. Here we can see an example of a molecular antibody that exhibits different attraction repulsion behavior under different solution conditions and its correlation to the extent of the aggregation when stored at 25 Celsius. This is a valuable observation at the early stage of developability, where you observe favorable solution conditions for a molecule with regards to the colloidal stability. And the observation might be able to guide us to understand which conditions, such as pH or buffer type, are most appropriate for the molecule. One very common stress applied for the biopharmaceutical candidates is thermal stress. During the developability assessment, the candidate molecules will be assessed for their thermal stability by incubating the sample at harsh temperature. This will provide a preliminary comparison of the degradation rates between the candidate molecule. For only one molecule candidate, it will provide a preliminary assessment of the potential route of degradation like aggregation or fragmentation using SEC result after thermal treatment. At the stage of developability assessment, material amounts are limited. So for most clients, through evaluation of the solution conditions that favor achieving high concentration is usually not feasible at this stage. One methodology that provides a measure of relative solubility is using a PEG preparation method. This method allows comparing the relative solubility for multiple molecular candidates and evaluating relative solubility in different solution conditions. At the lead candidate selection stage, there are several molecular candidates to be evaluated. This PEG solubility assay allows comparison between different molecules and molecular types as shown in the upper graph. Also, this method can evaluate one target molecular solubility through comparison with SBL internal database to understand the feasibility of the target concentration in the lower graph. So it is extremely important to be able to compare relative solubility during the developing period to select the most appropriate molecule for development. The PG solubility assay for determining the relative solubility can be employed to compare different solution conditions for a molecule. In this example, there is a clear indication for promising pH and buffer conditions for the molecule. This could be an option with limited material amount to evaluate solubility properties under different solution conditions. This is a case when we perform the real solubility testing with short-term holding time, stability, ambient condition to confirm the feasibility of high concentration and DSP process manufacturing. Our client wished it for 100 mg per ml, and we did stepwise concentration using the target molecule to confirm the 100 mg per ml. We successfully reached 100 mg per ml while retaining good viscosity of around 4 CPs. So we gave our client this data and performed the formulation development for them. However, I would like to say this study needs high protein amount compared to other scopes like thermal stability or biophysical characterization and relative solubility study using PEG. This graph shows hydrophobicity for various molecules. HIC analysis is not difficult, but the important thing is we can compare the results using our internal database to evaluate the molecular characteristics to predict the feasibility of process development. I would like to present on sequence-based molecular assessment using in silico program in Samsung Biologics. One of the first steps in the DEVELOPICK 3.0 is the evaluation of primary sequence analysis to identify hotspots for degradation in the amino acid sequence. For example, if there exists some hotspots for degradation in the CDR of the monoclonal antibody, then the formulation condition can be adjusted to account for those hotspots. So continuing with the hypothetical sequence, if, for example, there is NG, this might guide future decisions with regards to formulation pH. I'd like to introduce our own primary structure evaluation program. We can confirm these results like molecular weight, aromaticity, instability index, and gravity score using this program without uploading to the website, not only for maps, but also for complex protein like fusion protein or bispecific or multi-specific. We also can confirm that I already mentioned the degradation possible hotspots on the target molecular sequence using this program to prepare the formulation development strategy and DSP process development strategies. This program has already been analyzed for the commercialized molecule, where these results have been compared with website results, which were found to be the same for all analysis items. I want to share the case study using this program for fusion A molecule. As you see, we can calculate the molecule to know the potential risk like oxidation or deamination. And finally, we can confirm the risk that would help determine the optimal formulation screening matrix for the molecule. Another key advantages using this program would be that all cleavage sites on this sequence can be detected using previous collected enzyme cleavage site information with PI value. This information is very helpful to prepare the purification process or analytical method development strategy. So, DEVELOPICK 3.0 can select the best molecule among several candidates using small amount of protein within a month. Especially, the updated platform especially has the hydrophobicity measurement method and enhanced in silico tool. This tool will have even more opportunities to improve in the year to come. Thank you.
All right, the first question is this. So far, what is the experience that Samsung Biologics has had with the DEVELOPICK and its customers?
After launching DEVELOPICK in 2022, various molecular types were underwent the developability assessment program. As I mentioned, there were our clients who wishes to confirm their complex molecular process development feasibility for basic scope, which includes TOZ, TM irrigation, with short-term thermal stability and low pH stability. In most cases, the molecules were assessed developable, whereas several cases did not. I can say that with a specific case when we detect significant issues during thermal stability period, the molecule was multi-specific, and when we confirmed the thermal stability result, the molecule was deemed not developable in terms of stability. We reported this to client, and then was confirmed internal by them, as they found the material was made using an unsuitable buffer the molecule, which caused the molecule to degrade very quickly against the thermal stress conditions. After appropriate changes have been made, issues regards stability has been eliminated, and the molecule was developed in our CDO program successfully.
All right, another question. You mentioned that there is a seamless transfer of information from the DEVELOPICK stage into the process development stage. Can you elaborate a little bit more?
For programs where the material is generated for DEVELOPICK at Samsung Biologics, the upstream and downstream team are involved in the material production, thus providing the USB and DSP teams and on early assessment of the molecules. Additionally, as mentioned in the presentation, we also evaluate the low pH stability of the molecule. This not only helps to inform the formulation team, but also provide valuable information to the downstream team with regards to virus inactivation step, which employs at low pH hold. Finally, many of the developability studies are performed within a formulation development group, and often the same SMEs performing DEVELOPICK will continue to work for the formulation development for that program. Therefore, our SMEs can help effectively address the process development issues for the client molecule. the past observations of specific degradation pathways like aggregation or fragmentation will be taken into consideration for the selection of the various compositions.
All right, another question here. The biopharmaceutical field is very dynamic and competitive, where speed is often an important factor. Would conducting additional developability studies have an overall impact on the timeline?
I think that performing a developability assessment is even more important in the competitive and fast-paced world of biopharmaceuticals. One, it enables the selection of the correct candidate molecule to proceed into development. Additionally, the information gained from the developability assessment program helps to minimize the potential delays in process development for USP, DSP, and formulation. Especially from a speed perspective, in silico tool is very good for assessing the developability before starting the real process development stage, because this is not necessary using protein material or real wet testing, which takes time to confirm. Even though wet testing takes time to confirm the developability, these activities can be performed in parallel with other activities early in process. Therefore, this does not impact to the timeline since the material used in the developability assessment is generated by the transient expression system.